Yeah, so hello everyone. So I think it's working. If you don't know me, I'm Michael Grange, and I'm a group leader at the Rosalind Franklin Institute. And what I'm going to tell you today is a little bit about the work that we've been doing to characterize plasma focus ion beams for the use in structural biology and their wider applicability in various different workflows. I'm not here to sell you a plasma FIB. That's not my job. But what I do want to do is sort of frame the discussion around how we actually perform structural biology and electron tomography more widely, and how we can actually think about bringing together different modalities. So at the moment, we have various different hardware tools that we use to probe tissues and organisms on different scales. And at the cryo-ET side, we're really on that sort of structural scale. We're getting towards pseudoatomic structures quite regularly now. And so beginning to think about how we might be able to go back from that point to try and put as much context into those observations as possible is kind of what my group's been trying to establish and what sort of plasma FIBs are positioned to be able to do. But in order to do that, we need to think about why we're doing it, and the reason is to build up a complexity of approach. So we really need to start thinking about moving away from ease, where we've got 2D cell cultures. Cells don't grow in 2D cultures. And trying to build up that relevant complexity in that biological scale. And so at the Rosalind Franklin Institute, we have a few different tools to try and help us do that. So we have a Helios Hydra plasma FIB, which has lift out and a fluorescence module inside. We have a prototype Arctis microscope, which was developed with Thermo Fisher Scientific, and they gave a talk yesterday on their attempts to sort of characterize their workflows. And that has, an autoloader. You can have 12 samples. It has a fluorescence module, so you can begin to build up a fluorescence footprint of your cells. Then we also have a TEM, because obviously if we want to do correlative tomography, we need to be able to do some electron microscopy as well. And so just to sort of frame plasma, a lot of you probably used to gallium ion beam sources, and traditional gallium ion beam sources were sort of very useful for getting very fine probes to essentially etch materials in material sciences. But at very high currents, they start to become very divergent due to the nature of their of the ion beam of the ion source. So the source is a point source; it's very fine. So as you try and extract more ions from it, it becomes very divergent. Plasma was built in material sciences to essentially try and combat some of the issues with gallium, insofar as gallium can implant on aluminium, for example. So you can't actually analyze aluminium with gallium because you basically get these compounds produced. So plasma was an alternative ion beam source, and the nature of the optics there mean that it's harder to get to a finer point at low currents, like it is with gallium. But at high currents, you basically can have a very nice collimated beam, and the probe size remains relatively manageable. So it's really in that regime and that versatility with plasma that we want to get a handle on and start to try and characterize. And just for a bit of context, here's a slice of brain tissue on an electron microscopy grid. And you can see in the middle here a number of trenches which have been milled. And that's about 100 microns by 80 by 80 or so. And that took about 30 minutes with a 60 nanoamp current. And while that might be damaging, what it does allow you to do is start to shape tissue, so you can start to build up complexity. And if you start to use the scanning electron microscope, you can see here these tissues actually have quite a lot of contextual information. So on the left is some wild-type brain, and on the right is some muscle tissue. And so you can start to see various different things you might associate with brain and muscle tissue, such as myelin, synapses, sarcomeres, endothelia. You can see a red blood cell in this image. And so this really started off our attempts to try and characterize plasma. So different microscopes with come with different gas complements. Generally, it's xenon, argon, oxygen, and nitrogen. And so what we wanted to work out was for these kind of serial volume approaches, such as I just showed, which ion gas basically allows you to have sort of flattest surface for imaging, to mill at an appropriate rate. And so Maud started this work off where she basically wanted to know which gas should I use for my experiments? Which one's going to work out the best? And so she designed a way of trying to look at the curtains that are produced on the surface of a vitrified block. And then by using a Fourier transform to basically look at the lines that are generated, you can get an idea of the propensity of curtaining. So obviously you want a very flat surface so that your SEM images are great. And you don't want to have these curtains that are introduced. And what she found out was that argon had the lowest propensity for generating these curtains. And what we did was try to characterize the beam profile. So here on the right-hand side, you can see the beam profile as a function of current, so the size of the probe. And we tried to indicate the sort of shape of that beam. So you basically mill to a depth and you analyze the ion beam at that current. And you can see that argon has a relatively sharp probe characteristic. So it has this, this sort of relatively good puncture. There's a bit of a tail, that's why it's triangular if you want a flat one. But compared to the other ion beams, gas sources, sorry, this explains why actually with argon, probably that's why we get fewer curtains. But of course, a lot of us are structural biologists. So if we're using a FIB, we want to know, can we make lamella? So is the nature of the optics of plasma suitable for also generating very thin lamella for cryo-ET? And you saw yesterday, well, you know the answer already, but we also tried to use argon as well. So these are a panel of images where we show, you can produce nice lamella. They look relatively thin. They have, electron, you can see electrons through the lamella. But we wanted to see if we could do that in an automated fashion. So we did 3 different repeats and basically set up positions over 3 different sessions and want to know how many lamella can we produce, how many of them go through the different milling steps, and how many in the end allow you to get cryo-ET data. And you can see from the statistics here, it was 80% or higher for each of the individual experiments we ran. And we were fairly agnostic. We basically just tried to set up our positions. It didn't optimize too much. The other thing that we did as well was to try and work out what the milling rates of these different ion beams are in plunge frozen yeast cells. So it's all well and good saying that plasma is great, but is it slower or faster? Which gas source is the most appropriate for which application? And you can see from this list that the highest is xenon, which is about 17 microns cubed per nanocoulomb. That's how it's measured. Whereas the slowest is argon. So we actually used argon because we thought it gave the flattest surfaces and least curtaining, but it is actually the slowest of the 4 ion species. And just for reference from the literature, gallium is around 8. So gallium is basically about 66% faster than argon or so. We measured the thicknesses of our tomograms. So we took tomograms on all our lamella. I think we had, I can't remember the exact number, but we had a lot of lamella. And we took around 160 tomograms. And we, again, like I said, we didn't optimize for thickness. So here you have a range of thicknesses from about 180 to 350 nanometers. And this is taken from the data where we actually use particles, as I'll show in a minute, for subform averaging. The tomograms have nice cellular features, as one might expect, and as we've seen from various different speakers already. And we want to know, okay, are these data now suitable for pseudoatomic structure determination? Can we try and get information out that allows us to, basically say plasma is a viable alternative? So we basically picked ribosomes from our 160 tomograms or so. We had around 16,000 particles. We used WARP-M, as Sam just showed, to get a structure of the ribosomes from those HeLa cells. And we essentially were not ambitious enough with our collection parameters on the electron microscope because we actually got to micros. So we got around 3.8 angstroms in most of the 60S subunit. And as you can see, from the movie that's not playing, we can also see side chains in various different helices in the 60S subunit. So this was very satisfying. We basically were able to show that with plasma, at least we can get a structure out. But what was interesting was what's the effect of the ion beam on the damage to the sample. So, you know, you make a volume, you make a very thin 200 nanometer lamella, and then you take a tomogram, you have a three-dimensional volume. How close to the surface that you're milling should you use particles, and how can that inform decision making? Very thin lamella obviously have very little content. So if you're trying to do structural cell biology with a thin lamella, you might get very nice looking tomograms, but they're going to be very thin and not going to have too much in them. So having that play in thickness, I think, is very useful. And so what we tried to do was try and calculate the effect of the ion beam milling on the particles as a function of, perpendicular to the milling plane. So we basically did it in a shell approach. So we basically took 50 nanometer shells from the edge down to the middle, or 60 nanometers in. And we essentially calculated Savon averages for ribosomes in that shell. And what we were able to show was that the B factors of the particles from these different shells converged to around 40 nanometers or so. So basically, the particles from the center of the lamella had better B factors than the sort of global of all. So it means that those have the least damage, and those that are at the edge of your tomogram basically have some damage imparted from the ion beam milling. But that's not to say that they're not usable. It just means that you probably need more particles from that region to be able to use them or to achieve the same results. So plasma is a viable alternative. It imparts damage on the lamella. We knew that to some extent, and recently Bronwyn Lucas in Nico Gregoriev's lab did a similar analysis using 2D template matching for gallium. I think their value was 60 nanometers. So by that logic, argon has about 20 nanometer less either side damage imparted. And so with that, I just wanted to kind of talk a bit more generally about how we're using these approaches. So as I mentioned at the beginning, it's important to think about how we can actually gain context from our experiments. And so we're using these different modalities to try and actually target specific regions to put into our lamella to actually build up three-dimensional context. And so this is some data from a PhD student in my group who's trying to look at different Alzheimer's pathologies. And she basically overexpressed this variant of APP, which basically makes loads of vesicles inside go crazy. But what we just wanted to do was to see, can we test the fluorescence? Can we try to track different lysosome and different endolysosomal bodies in the cell? And so we milled, and then we can take a fluorescence image, and then we can use our serial FIB-SEM approach to basically build up that context. And I think what I want to basically say about this work is that, yes, a lamella is great, and a tomogram is fantastic, but if you want to get the full context of a process on a sort of whole cell layer, one lamella won't cut it, right? You need a bit more information to know what's going on. So it's really, it's really useful to combine these volume EM approaches such as cryo FIB-SEM with the fact that you can actually stop at some point and maybe make a lamella. And so you can see from these data, there are these vesicles. We can see, the nuclei in the top right. And what we're interested in is these blebby vesicles and trying to characterize those in a bit more detail. But you never know which plane in the cell they're going to be. So having the fluorescence combined with the SEM, combined then with the FIB-SEM to make a lamella, is kind of, a really interesting approach to try and do targeting. And so Maud has also done this. So we used Dave Farmer's sample, I think it's somewhere, rubrum, which is autofluorescent. And we're actually really able to track down through the fluorescence, through the autofluorescence in the fluorescence module, through the SEM, and then correlate those SEM images as we make a lamella to the TEM overview, and then acquire images of the septum of two bacteria dividing. And so we need to work out what the impact of such SEM imaging has, again, on damage. You are imparting electrons onto the surface, a lower energy, of course, but you know, it's concentrated because it's a scanning approach. And we also need to work out how can we actually automate this process, right? So if we're actually doing serial volumes, how do we know when to stop? Can we automate that process? And then can we make a lamella and do that in a way that's, you know, not very involved? Because, you know, people have better things to do than just to sit at the microscope all the time. I mean, I love it, but, you know. And then on the other side, so what we've also been doing is trying to characterize how we might be able to use plasma ion beams to image very thick specimens. So as I showed with the graph earlier, at very high currents, you can still harness a plasma ion source. You know, you can use those higher currents. So what we've been doing is playing with high pressure freezing to try and get brain tissues into different formats. So we want to basically, you know, make planchets, for example, for lift out, but also can we actually get these really thick specimens on a grid and mill them at various different geometries. And so Jake is a PhD student, and he basically has spent the past 6 months streamlining these freezing approaches. And together with Becky, who's in the audience, they have some nice fluorescence systems now to try and actually probe these tissues. So here are some fluorescent brain slices, which basically have neurons labeled. And we can target these within the grid and start to build up a picture of how the fluorescence is organized within that slice. And we can target a region, and we can mill it with a large current ion beam. And then we can make lamella. So it's quite involved. It's very thick specimens, but we can probably make around at the moment about 4 to 5 a day, as long as the sample is, you know, frozen well. And as someone said, you know, do we need a notch? For thick specimens on this order, so this is probably around 80 microns of material, you need a notch because it's basically going to go like that when you get thin. And it was really interesting because we made this notch and it was starting to bow, and as soon as you cut through, you know, it just went flat. So I think this notch is quite important especially for thick specimens, because there's, you know, basically vitrified ice is like a very, 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 very slow moving liquid, right? So it will basically concentrate pressure at the weakest point. But we can take SEM images, we can make a lamella, we have a TEM overview, and now we're starting to take tomograms on these lamella. Lift out, I mentioned lift out. So, you know, we can do lift out with gallium. So why bother with plasma? So if we just take the brain slices I just mentioned, right? So, you know, brain tissue is very polarized. You have dendrites and you have axons, and they go in different planes in Z. If you cut with a vibratome a brain slide, you know, a brain, you basically got 30 microns of cellular damage. So about 30 microns within that cut will basically be dying. A lot of will be dying cells because you basically just chopped them in half. So accessing sort of the inner regions of these type of samples so that we can actually gain lift outs, for example, that are the most contextually accurate is important. And so we've just been playing with it. We only got lift out on our Hydra back in January. So we've been playing with this a bit. And Jake again has been doing lift out. So we can actually now lift out tissues in a conventional way. But we can also do a little bit of, you know, fun experiments. So we can basically say, well, how big can we mill a piece of material for lift out? And so here's a 100 micron lift out. And I like to think of it a bit like taking a core sample from the Antarctic. You know, you basically have your brain tissue, and then you can basically just, you know, look at the striations. And we're also now trying to work out how we can use this in a correlative fashion. So here's just a very preliminary image of us trying to overlay our fluorescence with our lift out chunks. And so you can here see it make out probably like myelinated axon, which correlates with this red signal. And here you can see some Hush stained wild type mouse brain. So with that, I'd like to acknowledge my group. So Jake, Bronwyn, Ava, Becky, and Jess, the Rosalind Franklin tomography team. So Maud, who's basically been trying to lead a lot of these efforts. Tom, Casper, and Jim, who's the director. He also has a contribution. I'm sure he'd like me to say so, but yeah. And it's it's also important to acknowledge that this is, a very fruitful collaboration with Thermo. So the Actis prototype we have was developed over the past few years as a collaboration. And also our colleagues at Diamond, Sven and Jürgen, who we've worked on scripting with serial FIB for volume EM approaches. And then my collaborators on the right. And my lab's now funded by EPSSE and Welcome, and so is the ALS. And Maud has 2 job adverts out. So I made a QR code for her. So if you want to take a picture and have a look, feel free. And I'm happy to take any questions.